Thank you. So I'll begin with a statement that I think pretty much everyone can agree with, which is that depression and suicide are really serious clinical problems in adolescence that really stand in need of new insights and new ideas about what is happening in the brain and how can we advance our understanding of the neurobiology in order to guide treatment development, novel treatment development. Today I'll be talking to you a little bit about rigidity in depression and suicide. So we know that depression and suicide risk are associated with some limitations in problem solving, cognitive and psychological flexibility that have come up again and again in the neuroscience literature. This rigid thinking in depression, we see that patients get stuck on negative thoughts and negative mood states. They also experience, especially in that suicide risk period, this sense of narrowed options and the inability to imagine other alternatives outside of their situation other than self-harm or suicide, not being able to see a way out, and really a lack of a limited imagination in those moments of suffering. What might be happening in the brain with this inflexibility? The human brain's ability to flexibly change between states, neuroscience research has been telling us that this ability is critically important for adapting to the environment. And it's important for cognitive flexibility, having diversity of thought, and also for creativity. How can we measure it? Today I'll be sharing with you some results of resting state fMRI analyses, excuse me, and two particular metrics. One is looking at entropy, and one is looking at switching in between states. So let's say a little bit more about that. First, entropy. So we know that resting state fMRI, thank you so much, the signal fluctuates during the resting period, and we've been using that signal fluctuation for many years now and applying it and looking how brain regions connect to each other through functional connectivity analyses. But here we'll be talking a little bit about how to use that signal in a different way and measuring its entropy. So this is a concept came out of information theory, but entropy can be calculated from any time series data, and it's a measure of its unpredictability and its complexity. So basically, if you have a really highly regular pattern of a signal, then that would be low entropy, and if it's really complex, and it takes a lot of different bits of information to describe the signal, that would be high entropy, high complexity. And entropic brain theory would posit that signal complexity might underlie brain like cognitive flexibility and openness to new ideas and creative insights. There's different ways to measure entropy and calculate it, and in our work so far we've been using Shannon's entropy, and this is the equation. The other approach is through the use of dynamic functional connectivity analyses. So we know, and we've been aware in the field for some time now, that brain connectivity patterns are not static, but they are dynamic. And the extent to which these networks change over time probably has important meaning. And for example, it might signify flexibility in some ways. So for example, greater temporal variability of functional connectivity in brain networks has been correlated with flexibility, adaptability, and creativity in some studies. And other types of analyses, besides just looking at the extent to which the connectivity varies over time, can take the resting state fMRI data and characterize a series of brain states through which a person experiences during rest and goes between the states. And it turns out that analyses looking at frequency of state switching or transitioning between the states seems to be associated with cognitive functioning and creativity. So with that background, we looked at these two metrics of putative brain flexibility in three different samples of adolescents. Entropy, and the two metrics being entropy and state switching from dynamic functional connectivity analyses. So one of the first data set is about treatment-resistant depression in adolescents. It was a study where we did an open-labeled trial of intravenous ketamine, 6 infusions, and we measured these flexibilities before and after. The second one is adolescents with mood disorders, some depression, some bipolar. It was a trans-diagnostic mood disorder sample, and also healthy controls. And we looked at brain flexibility cross-sectionally, and also some of the adolescents returned to have a second scan 6 months later. So it was a naturalistic follow-up. Third, we have a sample of adolescents with non-suicidal self-injury, which we know is a significant risk factor for future suicide attempts. This is a project that is a longitudinal project that's ongoing, and it's a preliminary data analysis from that study. In terms of the methods, for sake of time, I will just say that we were able to use state-of-the-art acquisition and processing technology thanks to and adapted from the Human Connectome Project. For the entropy calculation, what we did was we extracted bold signal time series from limbic regions. We focused on the amygdala, nucleus accumbens, and subcallosal cingulate. And we performed a wavelet transformation, and then we calculated Shannon entropy in each of these regions of interest. For state switching, what we do is there's different ways to do this. We utilized the GIFT Dynamic Functional Connectivity Toolbox. So this is a tool that was developed by Vince Calhoun and his colleagues. So you start out with an independent components analysis, and you identify networks that look like real brain networks of interest, and enter them into a sliding windows analysis. And this generates 848 basically windows of correlation matrices. And those then we perform a k-means clustering to those many correlations, sliding windows, and then that gives us our states. We used a K of 5, and so we had 5 states, and then we measured the frequency of transition between states for each person. So for results, the adolescent ketamine study, our key finding that I'll share with you is this correlation between improvement in depression and entropy change. So increased entropy after ketamine was associated with clinical improvement. So on the y-axis, you see the CDRS % change. So in this case, if it's higher, that means you got better. And so these are our 5 responders in the top right corner, and they had a big change in the right nucleus accumbens. And then here's our non-responders. They really didn't change in their entropy at all. And this was a similar pattern across regions that we looked at, but it was most significant in the right nucleus accumbens. For the adolescent mood disorders study, we also looked at entropy in these regions cross-sectionally, and here we found an inverse relationship between entropy and depression severity. So higher depression, lower entropy, and it was again more significant with the nucleus accumbens, but it was a similar pattern across regions. And interestingly, at follow-up, an increase, this was not associated with the treatment, but just spontaneous increase in a clinical improvement was associated with increased entropy in these regions, but it was most significant for the amygdala in this case. Next, we looked at state switching for the adolescent mood disorders study. We found that, so we were thinking, well, entropy is one measure of flexibility. What about this other measure of flexibility? And we indeed found that higher depression symptoms was associated with fewer states. switching. So tending to stick in one state more throughout the resting scan. Third set of results, adolescents with non-suicidal self-injury. Here we did find the mood disorder study, we didn't have group differences with controls, we just had the correlations primarily, but here we did find a group difference between girls with non-suicidal self-injury versus without, where the girls with non-suicidal self-injury had lower entropy, and it was most significantly, in this case, in the amygdala. We had these findings with the subcallosal cingulate in this sample. A similar pattern as we had seen before, inverse correlation where higher symptoms are associated with lower entropy. And we found it with both depression scores and also suicidal scores. Is it time? Yeah. Oh, cute. And this withstood correction for, the suicide finding was still present when we corrected for depression symptoms. Similar finding with the other regions of interest. State switching with the adolescents with non-suicidal self-injury. It was a very similar finding as the adolescents with mood disorders. Higher depression was associated with lower state switching, more tendency to stay in the same state. Finally, we looked at the correlation between state switching and entropy, these two metrics of brain flexibility, and they were positively correlated across most of the regions of interest, especially right nucleus accumbens. So in summary, in 3 different studies, we found preliminary evidence for entropy and state switching as fMRI markers of neural flexibility related to depression and suicide thoughts. Some possible evidence that these 2 markers, putatively of neural flexibility, are related to each other, or coherence across flexibility measures, and some suggestion in small numbers of neuroplastic changes associated with recovery. So in conclusion, brain signal and network configuration complexity has been associated with mental flexibility and creativity. We're interpreting our findings as possibly meaning that this is underlying a propensity to get stuck on negative thoughts and having difficulty seeing alternatives to suicide. And finally, we feel that these preliminary findings, if confirmed, could suggest a potential opportunity for pursuing this as a treatment target with interventions designed to boost neural flexibility in adolescents with depression and suicide risk. Thank you.
We have time for 1 or 2 questions.
Yes, have you considered looking at performance on creativity-related tasks? There are a number of tests out there to see if those are orthogonal to those responses or related.
That's what we'd like to do next, yes.
So a bold time series is typically continuous data, and chain entropy is calculated on discrete data. So did you discretize your data somehow, and if so, how did you do it and why?
What was the bold is continuous, and what was the second thing?
But Shannon entropy is calculated on discrete, so integers. So how did you discretize your data?
We didn't. We just entered this. This was the. We used a function in MATLAB. I don't know exactly how it works.
The one that's meant to process images?
It's called Wentropy.
Yes. Okay.
Yeah.
Cool. Thanks.
Sorry for not having a better answer.
So I think those of you who have additional questions can approach Kathryn afterwards. Thank you, and let's thank her for her really wonderful talk.